Welcome, everyone. Welcome to the third live discussion. So, today we'll be discussing more about the week three contents and some sample questions from week three, which were in the previous assignments. And then we'll have a discussion on all the questions. So, due to some technical issues, we'll have for 40 minutes sessions, 3 40-minute sessions. Once the session gets over, please rejoin the same link within a minute. Okay. So, let's start. I'll share my screen and we'll just proceed with the discussion. Once I share the screen, just somebody confirm if my voice and my screen is properly visible or not. Okay. So is the screen visible and my voice is also properly audible?
Yes, ma'am.
Okay. So we'll begin. I hope you all had a very good time attending the live session that Professor Srivastava conducted last week, and you had an idea what kind of work they all are doing, or the kind of work we are doing, and our respective roles. So, uh, and and I hope the rest of the course is as enriching as it is. It seems so. Okay. Okay. So before we discuss today about some questions, I would like to discuss some few topics or few concepts that I feel that all of you need to understand. The first thing is that as per this week's content, we are already done with different kind of vectors that we've discussed what vectors are, how cloning is done using vectors, and what are their roles. But the kind of gene that we are expressing or the kind of different proteins that we genetically clone and derived from bacterial cells, many of them come from eukaryotic sources. So the problem is that eukaryotic proteins are quite different to that of the prokaryotic proteins, like the ones that are grown in bacteria. So any gene or any protein that is within a bacteria or under bacterial promoter regulation will follow all the rules of replication, transcription, translation using the bacterial promoter. But if we have to incorporate a gene which will produce a protein, and that too that comes from a eukaryotic source, or a particular gene from any eukaryotic source into a bacterial cell, then the story slightly changes a bit, because there are few points that we need to remember. One is that eukaryotic proteins could be toxic for the bacterial cell due to many reasons, like high production, or due to the chemical makeup of it. There could be different reasons for it. Also, in prokaryotic system, if we look into the central dogma of it or into the process of translation, the entire eukaryotic DNA or the prokaryotic DNA is quite different. Like the RNA is quite different. So once from DNA the RNA is formed, there are areas called exons and introns. So exons are the regions from where the protein is translated, where introns are deleted, and this is exclusively present in eukaryotic system. But in case of prokaryotic system, there is no such thing of intron or exon. There's no such discrimination. Whatever is the RNA, from there we get the protein. Like from there translation occurs and we have the final product, the protein. So if we are putting a piece of DNA within a vector that is of bacterial origin, and also it has a promoter that is recognized by the bacterial polymerase, then how will it understand that this is the exon and this is the intron? It will actually try to translate the entire thing. So this particular issue is actually overcome by inserting specifically those portions of the RNA or the transcript which will only have the exon and not the intron. So that's how it's synthesized, that we have the DNA that's called the complementary DNA or the cDNA, which has only the coding sequence. So what is a coding sequence? A coding sequence is nothing but a compilation of the exon, which will ultimately be translated into the protein. And the non-coding sequence is the intron. So if we look at this picture in the right side, we see that this is a eukaryotic DNA. It's a DNA, it's a promoter region. It has an exon. it has an intron, and like this alternatively it has exon intron. Then after transcription, again this exon and intron is transcribed, and this is known as the primary transcript or the RNA, the primary one. Again, there's an event called splicing, which actually removes the introns and keeps only the exons to it, and that exon is ultimately translated into a protein. So what is done is this RNA, which has only the exon, is assembled after splicing, and using a reverse transcriptase, a reverse transcriptase, what does it do? It is again this kind of a polymerase. Generally from what happens is from DNA to RNA. But if we want to get the DNA out of an RNA, then the role of reverse transcriptase come. It's a reverse of transcriptase. What does normal transcriptase can do? That is transcription. But if you want the reversal of it, so from an RNA to a DNA, and that DNA is the coding sequence. Why? Because it came from that particular RNA which had only the coding sequences or the messenger RNA or the exons. So the DNA that we have after it has been reverse transcriptized, it is the cDNA or the coding sequence. This is the gene of interest that we insert into the plasmid. So what we discussed, or what was discussed in week 3 was, week 2 was, we insert a gene of interest within a suitable vector so that it can be transformed into a different host, and then we can have a gene of interest in it. Okay, we can have a gene of interest in it, amplified gene of interest. So here directly we don't insert the gene, but we do a bit of we do a bit of engineering in it. And what do? We take the cDNA and we directly insert it into the plasmid. And these particular plasmids in which we are actually inserting genes which will ultimately give us proteins, and that also which are of eukaryotic, the gene that's come from a eukaryotic source, these plasmids are known as expression vectors. Why? Because ultimately our aim is to have the expressed protein or the gene of interest in that sense. So it is an expression vector. And there are some few even in case of vectors, there are few categories or few things that we have to so that it's termed as a good vector or a proper plasmid. So what are the different points that an expression vector should mandatorily have, otherwise it cannot be used? One is a ribosome binding site, a terminator sequence, a strong regulated promoter, and also definitely we cannot forget that the eukaryotic gene which is in the form of a cDNA, the copy of the mRNA. Now one by one I'll come that to the importance of all these 4 points. One, and apart from these 4 points are the main thing which are mandatory, and apart from it we'll definitely have one antibiotic resistant gene which will help us in screening or selecting the colonies when we are transforming it. That is also important. So let's come to one by one why these are used. First is the ribosome binding site. So ribosome binding site is it's a particular site in which the ribosome binds, as the name suggests, and from where it decides that from which point the translation will begin. We know that in translation, the ribosome binding site plays an important role, and it actually decides that, okay, from here, the event of the translation will begin. Terminator sequence, it cannot keep on translating the entire gene again and again. So we definitely need a terminator sequence, because suppose I say this is my gene of interest, like I'm showing it with the help of my cursor. So if my translation starts maybe from here, from the red region, if I don't give it a signal to stop, then the entire thing will be translated. But I only need this portion, which is my gene of interest, which will give me my protein of interest. So I definitely I need to say, okay, now stop. Once you're done translating this part, you need to stop. For that, we need a proper terminator sequence. So what is a terminator sequence? A terminator sequence is a sequence which tells it to stop. It's basically the 3 different codons that we use in a normal end site, that we say the end site of a translation. Third point is very important. It is a strong regulated promoter. Now what happens is a bacterial cell is small in size. Now if the protein of interest is continuously produced, it will start accumulating and there will be space issues. And also it can be toxic for it. So initially we need our cell to grow and divide, and then it should start like producing the protein of interest, so that these two theories will not clash with each other. And as a result, we should have a very strongly regulated promoter which we can control from outside when we want. Like if I want this protein to be transcribed now, then it will be transcribed, and if I don't want it to be transcribed, then I will stop it. And these two signals exactly happen in a switch on or in a switch off manner. And there are repressors, activators, and these signals are transferred in the form of an inducer, that an inducer will actually say, okay, now the translation can begin. And again, a repressor will say, no, the transcription should stop, and it is not required now. Okay. And the third and the fourth very important point is the eukaryotic gene, or which is a cDNA copy. We have already mentioned or discussed that what it actually happens, like why we need a cDNA and not a normal DNA or a RNA. Okay. So the most important feature, as mentioned in the slide, is the tightly controlled promoter region. So we can have the regulation in our hand, and also like these the kind of induction signals that I was discussing. It could be anything. It could might be a chemical, it could even be temperature. Like there are certain vectors in which it's designed in a way that at a particular temperature, if that signal is given, like maybe normally we are growing it in 37 degree, and maybe we increase it to 40 degree or 42 degree, at 42 degree that particular gene will be expressed. But if even a lesser or a higher temperature is given, it will not be expressed. So that's how this inducer signals work. Now this is the basic overview of expression vectors. Now, why am I discussing this? Like, why are expression vectors important? Like, okay, now I have included my gene of interest, which is the cDNA. It will give me a protein. Now, not just one protein, but different proteins will be produced by that particular organisms. And it's not just one organism, it's multiple bacteria producing that particular protein in your culture. And there, so the end product will be a complex or a mixture of different proteins. But how will you isolate your protein of interest from that mixture? For that reason, we specifically use a thing called, we actually use, what we do is we engineer it in a way that we add tags to that gene of interest. Now, till now, we saw that there's a promoter, there was your cDNA, and there's a terminator region. So what we do is there are different kind of tags that are available. We add that gene of that tag to this clone beside the cDNA. It could be on any side of the cDNA. It could be on its left or it could be on its right, depending on which terminal we wanted. Like it could be N terminal or it could be C terminal. So at any point you can like add this six. It's a histidine tag. It's generally a six histidine tag, and it has six histidines on it. Like it's a polylinker kind of thing. So once your transcription and translation is over, you will have your protein in this way, that one end of it will have 16 tags. Now this histidine bind, now now it has a histidine tool. Now how will we purify the protein that has a histidine tool? We will add this protein to a, we'll try to isolate this protein by adding it to a like to a column which will have something that binds to this histidine. So when you're passing your solution through it, only the proteins which have those tags will bind to that column, the whatever it's present in that column, and the rest of the proteins which were not of your interest and did not have the histidine tag will not bind to it. Now when I'm talking of binding, that histidine will bind to something that is there. It could be either an antibody or it could be a nickel. In case of nickel, anti columns, nickel forms a coordination complex with this histidine. So that will bind to it and the rest will wash off. And again, once everything is washed off, we'll give a elution buffer with which the histidine, the protein that was bound to it, will break, and then you can collect it. So that is how we do the purification technique. So thinking of something way way like ahead of the process. Like it's not just you just have to clone something, you have your protein and your experiment is done. No, you also need to isolate the protein in a purest form. For that, to serve that purpose, you are designing it in that way. Okay, I'm cloning my protein, I'm attaching a tag to it so that when I'm trying to recover my protein, it gets easier out of all. right? So what is this tag? It's a small tags can be fused into protein of interest using the expression vectors. These tags will allow the protein of interest to be isolated and pure. So if we see that now this histidine is tagged with this protein. Now if you see, this is a mixture of proteins, and these small ladder, like it's actually H, it's written H, but it's looking like a ladder in this picture. So these are actually the mixture of proteins, and the yellow ones are your protein of interest, which has these histidine tags. Now you pass it through a nickel column. Now proteins are poured through the column, and what happens is, you see the nickel column is binding to this histidine. The his tag proteins are bound to it, while the rest of the proteins are passing through. Now you like elute it with histidine or imidazole, and then the tag protein is displaced. Now these histidine and imidazole will actually act as a competitive binder to it, so inhibitor to it. So what will happen, because it has to bind to histidine, it will take the histidine along with it and displace it from the nickel column. That is how we are doing. So till now we have discussed the expression vectors and the tag system. So is it clear to you, to all of you, that how
Yes,
this
ma'am.
histidine tag works?
Yes,
Okay.
ma'am.
Okay, so keeping this in mind, let's try to solve one question which has something related to it. Okay, come on. We'll go through the question together. So in this particular question, just give me one second. Yes. Okay. So there's an MSc student who is working on a human gene, that is insulin, and he wishes to produce the protein for therapeutic purposes. So we need to help him to purify the protein, where he has already used N-terminal histidine tag sequence to facilitate the protein purification process. So when this particular MSc student is doing this experiment, so he need to choose a correct expression host. So out of these four following features, what particular point should he keep in mind before like designing his experiments? So option one is length of the gene, like length of the uh, gene of interest, presence of introns that we discuss, introns and exons. Third is post-translational modifications, and that we were discussing that the proteins are quite different. By post-translational modifications, what I mean is, suppose we know that a protein that is translated is not the final version of the protein. Different kind of modifications is done to the protein, and then we have the final form. Now out of these modifications, something is known as post translational modifications, like it could be addition of a carbohydrate, a glycosyl group, or a different kind of moiety to it, and it gives a final form. Now post translational modifications are quite different in the eukaryotic and prokaryotic system. So if we are trying to clone a particular protein within a bacterial under a regulation of a bacterial system, but the protein is of eukaryotic nature, the complete post-translational modification getup could change, because it is ultimately under the control of bacterial system. So the correct moieties or the correct groups will not be added. Maybe something else will be added, and you will not actually get the final form of the protein. You will get a protein, but that is not the protein of interest that you want, because slight addition in one moiety can actually change the entire function of a protein. So the role of a protein in the downstream process, that for why you need it. So that is what is meant by post-translational modifications, that uh, it's different in both the systems, eukaryotic or prokaryotic. And the final option is the toxicity to the host cells. So what according to you could be the answer, like out of the four, what could be, what point should this particular MSc student keep in mind before designing the experiment? I have one thing, something in the chat box. C, okay.
Ma'am, post-translational modifications.
Okay. And rest of the students? Good. C. So I've already discussed what I mean by post-translational modification. Yes, that is the correct answer. But I would like some of you to actually elaborate on it, so that I understand that it's clear to you that why post-translational modification is a very major event in it. Provided that we have thought of, okay, these things are not there. Because when a person is actually trying to incorporate an eukaryotic cDNA within the bacterial system, he or she has actually uh, designed the cDNA, not the normal DNA. So the presence of intron concept is gone. Length of gene, yes, we know which vector can accommodate the like infinitely long DNAs cannot be incorporated within a vector. This concept is kind of conventional, it's known. So whenever somebody is designing a vector or a gene of interest, he or she keeps in mind, okay, this is the maximum length that I can incorporate. And the fourth point, toxicity to the host cells. It is known that particularly this protein will not be toxic to this, and there are measures of like taking care of it. So the third point, that post-translational modifications is the most correct answer here, or is the only correct answer here. And I would like one of you to just slightly say that what you understood by post-translational modifications and how it is important or significant for this answer. Any one of you? Anyone? I see 9 participants. Any one of you can just unmute yourself and just elaborate or just show some light on it. Okay. So I guess this concept is clear, like post-translational modification, addition of required marker. Okay. Yeah. Thank you. I thank you for just typing it out, but I would like really appreciate if you just voice out your points also. That's fine. Okay. Let's move
Ma'am,
on to the,
after
yeah, yeah.
translation, that means the production of proteins, there are some changes which take place, and these are post-translational modifications. So, in this reference, because we will be detecting our protein using the histidine tag, that's why it is important that we choose an expression system with post-translational modification.
Okay. So as you mentioned, there will be some changes. So when a particular protein is translated, is that the final form? Like, is that protein the final version of the protein that you want? Or what is the requirement of this post-translational modifications? Like, what would happen if any protein was not post-translationally modified?
And it modifies the protein so that they can function properly.
Yeah,
Uh,
correct. Correct, correct. Okay. So somebody is asking me that, ma'am, can you also please elaborate if the length of the gene matters? Yes, definitely it matters because if you in week two, I guess when we were on the very preliminary stage when we were discussing about vectors, if you remember, there's a concept that the length of the gene that you're inserting cannot be infinite. Like obviously, if it's very big, even bigger than, you are removing a part of the gene and you are inserting a gene in that place. So these two things could match. It's not that you're just opening a vector, putting something and leaving it for transformation. No, generally we remove a part of the vector or the plasmid and we insert something in that place. So if that gene is greater than what is being removed, then definitely the entire system will collapse. It cannot, like, there's a time for replication. It doesn't happen in that way. So yes, that is also important. But in context of this question, length of gene is not important because he's already decided that he has an N-terminal his-tag gene. So somebody who has thought of something so ahead of it, I hope that this person has already thought in mind ki, okay, my gene length must be within this range. So yeah, that is important, but not in context of this question. Any other questions from this concept? You can actually come up with any kind of question that is related to this, like not just specifically to histidine tags, anything that is related to expression host or vectors, what kind, like anything. I'll like wait for 1 more minute and see if we have. Okay. So I'll go with the next question. Hmm. Okay. So again, he has selected a particular PCR confirmed colony of a transformed P. lyse bacterial cell for protein expression and purification. He lysed the cell, extracted all proteins, and desired and the desired protein is histidine tagged. So, which of the following chromatography technique will be useful for protein fixation? The first two lines we can just forget for the time being. Just remember that this particular master student has purified a particular, he's actually picked up some colonies which has a particular strain of bacteria or a particular vector in it, and he knows the gene of interest in it, and his gene of interest, which will be ultimately translated to a protein, has a histidine tag to it. And after lysing the cell and extracting the entire all proteins from it, he has a complex of proteins within which his gene of interest or protein of interest is there, which is again tagged with histidine. So now he has to purify it. We know that now we are isolating the proteins from the nickel bead columns. Whatever is done is fine, but now still on we need a much more purified. Sometimes some other proteins stay in. So which chromatography technique will be used for protein purification? Like, what is the choice of chromatography that if you were given such a problem or you were doing something in the laboratory, what you had done? So just anyone try to attempt this question.
Ma'am, affinity
Uh,
chromatography.
Good.
And because histidine will bind to nickel beads, because it has a good affinity,
Hmm. Hmm.
good binding affinity, only that it would be able to bind, and hence it is the most appropriate. I mean, it is the only one which we should take here, the chromatography technique.
Yes, sure, sure. That's great. Huh. That's absolutely correct. So that is the concept of chromatography or the main motive behind using a histidine tag. Like why we are designing it in such a way that it has a histidine tag so that ultimately we can purify using this. We can actually exploit or take advantage of the fact that okay, it has a histidine tag to it, so I'll subject it or make it pass through a column which is something that binds to histidine, and then all the proteins which has histidine tags in it will bind to it, and eventually we can elute it. So just for all of your references, we will just briefly discuss the different chromatography techniques that are mentioned in this, in like the options, so that we are able to understand what exactly happens in a chromatography. So in a very brief way, a chromatography is actually a separation between two immiscible liquids. Okay, now it's not just liquids, between two phases, I would say, not exactly liquid. So what happens is you have two phases. It could be liquid or gas, and there's one more phase which is mostly stationary, and it could be at times mobile also. What happens is the mostly used chromatography is a column chromatography. So in a column chromatography, there's a column present, which is actually the stationary phase. And over it, we pass the sample to, which is the mobile phase. Now, this mobile phase also has buffers in it. It's not purely the sample, but it has buffers in it also. The entire sample will pass through the column, and depending on different properties, like it could be electrostatic interactions, size, or affinity, the samples get eluted or are retained within the column. So, in very brief, this is how a column chromatography looks like. As I mentioned, there's one phase which is the stationary phase. This is present in exactly within the column. It is mostly a solid and a porous matrix kind of a thing, which has it's made up of different kind of polymers depending on the type of chromatography that we are selecting. We'll discuss that what happens. So here is the column, and here is the reservoir. So this reservoir has the protein sample in it mixed with the buffer. So you pass your sample through this and it percolates through this column. And then you have your eluent. So if we see, irrespective of the fact that what kind of column chromatography this particular picture denotes, let's see what is actually happening. So you have your sample, you see different colors are getting separated or resolved. And here even more, and we see the green color sample is coming first and the red one later, followed by blue and the gray. So you see that there must be some physical or chemical property of this column that is making the proteins present here elute in a different way, and you see the type of elution or the resolving power is also different, like which sample will be eluted first, which will be different. So you can just, in case of this kind of a column, you can just collect the different colored solutions, and you have the four different, maybe the sample had four different proteins in it. So 4 proteins are colored in 4 ways. So these 4 colored solutions will have your different proteins in it. So that's what happens within a column chromatography. Now, this is just brief thing, like overview thing, but yes, we have it according to our purpose. Like what kind of column are we going to use will depend on the type of experiment we want. So the very first one here, it is the it is okay, sorry. So this will be the size exclusion. I've just missed the name here. This is the size exclusion chromatography. So in case of size exclusion chromatography, what happens is, it is the exclusion criteria happens on the basis of size. So what is there? The sample has different kind of proteins, some larger size proteins and some smaller size proteins. So what happens is that you have a stationary phase. This is the ones which are yellow. These are porous polymer beads, which has pores within it. So once, and these pores have specific sizes too. So suppose you have a sample which has different size proteins in it, and you pass that sample through these pores. Okay. What will happen is, the the proteins which exactly fits the size of the pores or even lesser than it will go through those beads. But the proteins which do not fit within it or are larger than the pore size will just move through it in a serpentine fashion. Like you see, there are gaps between the pores. The larger size proteins will just elute like this, and they will not go any within any pores, and they will be larger size proteins. And so larger size proteins in a size exclusion chromatography elutes faster than smaller size. Why? Because smaller size gets trapped within the pores and they actually elute on the basis of their size. Right? So here we see that it's given for 6 different tubes are kept below it, and it says that protein molecules separate by size. Larger molecules pass more freely and appearing in the earliest fractions. Like if this is all the fractions in it, this one appears earlier. Correct? So is that clear? What is a size exclusion chromatography? And what is the principle behind it? That why size is a factor. Different size proteins, you pass it through beads which are specific sizes. Smaller ones get trapped within it. Smaller ones in the sense that the proteins which have size equal to that or even smaller than it. And the larger size proteins which cannot enter the porous beads will easily pass outside it or from the outside, not getting within it on a serpentine faction manner, and it will elute first. Clear?
Yes, ma'am.
Okay. So let's talk about the another kind of protein, another kind of chromatography that is actually very of interest in this. That is the affinity chromatography. By affinity, we mean that whatever we are trying to elute or we are trying to isolate has something related to affinity. So we make a column which has like a ligand which will bind to our protein of interest. Okay. So what happens is, this is a protein mixture is added to the column containing a polymer bound ligand specific for protein of interest. We put the protein mixture in it. And then what do? We just, let me just basic first say, it's very clear, it's very easy that first we have a column and it has some tags to it within it or some complementary binding agents to it. Now, if we subject or we pour our protein through the column, the proteins which have the tags which bind to those particular things that are already within the column will bind to it, and rest of the proteins will be washed away. Now, this washing thing is actually done with the help of a buffer, wash buffer. But it is okay, okay. Protein. Okay, sure, sure, sure. There's a question to week 3 assignment, question 9. It's about, can you just copy paste the question in the chat box, Prachi? Then I'll get back to this question right after discussing this. Okay. So what is done is, the first thing, that a mixture of proteins is actually sent through it. And you see the ligands are bound to this, the beads that it already has. For suppose this was the histidine nickel column. I'm saying just for example, the histidine nickel column. So the protein here has histidine, the mixture of proteins here has the histidine bind to it, and the ligand here is the nickel. So we we pour the protein solution over this column. Then we just do a normal washing. By washing, what I mean is that using a wash thing so that unwanted or the extra protein that doesn't bind to the columns will get washed through it. Okay. And then we again, so they are getting washed through it. Why? Because these mixture of proteins are already bound to these ligands. They have already bound to this ligand, so there's no other place left for the unwanted proteins to bind. So it will anyway get washed away with the washing solution. Then we again add a solution of ligand to it. I said competitive inhibition. What it happens that it will dislodge or displace the histidine bound to that ligand, and the ligand will itself bind to it. So what happens is you see the protein of interest is eluted by the ligand solution. That is how you extract. Now, what is the thing that histidine attack proteins are already bound to the nickel, but somewhere or the other you need to like take out those proteins. Ultimately, you will require that proteins, right? So for that, you need to supply it with one more round of solution which will have the ligand, and it will actually remove it. Remove it how? The ligand will competitively, the ligand, sole ligand will competitively uh, come into a competition with the ligand bound to the post so that it is able to acquire the histidine and bind to it. And that is how the histidine will bind to this ligand and will be eluted. So if we see the picture, this is the protein of interest in a crescent shape and it's in a moon shape and the ligand is red. So you see here, this histidine, this particular histidine bound protein, this one, if it's visible, I don't know. Yeah, this was bound to this ligand. Now, on I'll just start within 2 minutes once. At least we have five, six participants. I'm really sorry that chat got disrupted. Okay, so I'll just. Okay, so we were here. So you see this, the beads. Is it visible, everyone?
Yes, ma'am.
Okay, okay, okay. Okay. So you see, these beads had these ligands to which the protein of interest was bound. Now, in order to break this and have only the protein, you again supply this with another round of ligand solution. What will happen is that the ligand will, both the ligands, one bound to the polymer beads and one which is not unbound, will compete within each other so that the unbound one gets the histidine proteins to it. And that is how they bind to it and then we elute it. So is the concept of size exclusion chromatography clear?
Yes, ma'am.
Okay. Now, now another form of chromatography which is known as the ion exchange chromatography. So the term ion exchange, it's like if you see actually these chromatography, the terminology itself guide you through the process that what kind of thing is that. So ion exchange is again you if you actually observe the end all the kinds of chromatography, mostly it is depends on the kind of like how will a particular protein bind to a column or be retained within a column and how are going to elute it and which is not binding. And that will depend. The ones which binds to it and takes time will elute later. And the one that doesn't bind or has nothing to do with the column will just elute like that. It will just flow through. It will not be obstructed at any part of its flow. Okay. So here in ion exchange, what is this? What happens is there are two kinds of ion exchange. One is cation exchangers and one is anion exchangers. So exchangers means that they have a particular charge and they attract a oppositely charged ion to it. Like cation exchangers are generally negatively charged. So if something is negatively charged, it will obviously attract something positively charged like cation exchanger. It is itself negatively charged because it will attract cations to it, which is positively charged. That is how it happens. That's the concept that how it is happening. So a cation exchanger is negatively charged and it binds to a cation. Similarly, an anion exchanger is positively charged because it is attracting anions to it. So now if your protein has positively charged, it has positive charge to it. So what kind of exchanger would you use? Suppose your protein solution has it's positively charged. All your proteins are positively charged. So what kind of an exchanger would you use? You may use the chat box, all of you. Your protein is positively charged.
Ma'am, cation exchange.
Good, very good. Why?
Ma'am, it is negatively charged and attracts positive towards
Exactly.
it.
So because your sample is positively charged, it has to bind to something which is negatively charged, and that is a cation exchanger. That is how it happens. So if you see in this figure, what happens is this beads are negatively charged. So it's actually negatively charged means it has negatively charged functional groups attached to it. That's how the negative charge is imparted to it. And then it you pass a solution of protein that has different kinds of charges to it, and you see which kind of charge will come first. So proteins will move through the column at different rates. Now, charge of proteins is actually determined by the pH. So you are aware of the concept of isoelectric point, like at particular pH where it's actually neutrally charged. Below it or above it, it has different charges to it. And that depends on the buffer or the kind of pH that you're providing to it. So depending on that, charge of protein changes, and then you can actually separate your protein. Now this buffer has the different pHs. Like you apply you have a protein solution, you try to elute it with a particular pH, the protein will get that charge, and then accordingly it happens. So is it clear, like uh, what happens, like what is happening? So
Yes,
just tell
ma'am.
me, if you have a cation exchanger, okay, you are using a cation exchanger, that means your positively charged proteins will get attracted to it. So what? So will the and you have a mixture of proteins, like both positive and negative charged proteins. So when you are eluting it and you are analyzing your elutions, what will happen? You will get the positively charged proteins first or the negatively charged proteins first. What will happen? You want me to
Ma'am, will you
repeat?
please repeat the question?
Yeah. So suppose you are using a cation exchanger and you have a mixture of proteins. Okay. So which kind of proteins will come first? Like your positively charged proteins or your negatively charged proteins?
Negatively charged proteins, because the positive charged proteins will bind to the cation exchanger. So negative charged proteins will come out first.
Hmm. Good. Very good. So is everybody at par with her or somebody has any contradictions? Does everyone agree with her?
Yes.
Okay. Now we'll discuss the fourth kind, which was in the question. It is slightly different to this column chromatography. Ultimately, it's a column chromatography. There's a column which is stationary phase and you mobile phase, that is same. But here you pass your samples with high pressure. High pressure because this pressure will actually enhance this process in terms of time. And this kind of process is again coupled with the mass spectrometry. So in combination with that, high pressure is used. Like here the samples, you're just pouring it and letting it elute as per it is its own pace, but you want it to happen earlier, so you just forcefully give a particular pressure and you subject your samples to the column exchange. Okay, so that is the normal phase chromatography. It's a sample, you see this phase which is stationary is a polar phase which is made up of silica, and your sample is passed through a non-polar phase which is non-polar. That is a normal phase. And there's one more phase which is called a reverse phase chromatography, in which your sample mobile phase is polar, but the stationary phase is non-polar. So why has this, now this is the most used process, reverse phase, because most with time it was seen that major of your samples are non-polar. Depending on the polarity of the sample, we decide that which kind of it will be a reverse phase or a normal phase chromatography that has to be used. So just I'll go back to this question once again. It was a very simple question, but I just thought of elaborating the different chromatography techniques with all of you. So in this context to this question, definitely we have to use the affinity chromatography. So is the concept of chromatography clear to all of you? At least the four different techniques that we discussed. Is it clear?
Yes, ma'am.
Okay.
Ma'am, will you please explain the reverse phase chromatography once again?
Okay. So what happens is reverse phase on the basis of your sample, like what is the polarity of your sample, we determine what kind of phase we'll be using. Okay. So what the same concept. If your stationary phase is polar, so non-polar substances will get it's like stationary phase is polar, it is made up of silica, and your sample, the mobile phase, will be non-polar. But if you are using a reverse phase, the polarity will just change. Like mobile phase will become polar and the stationary phase will become non-polar. Now, how do we determine that what kind of phase we have to use, a reverse or a normal phase? So just for very in a basic way, if we talk about it, it depends on the polarity of the analyte or the type of sample that you want to analyze. That will determine that what kind of polarity that will determine what kind of phase of chromatography you will be using. If you is that briefly clear to you?
Yes, ma'am.
Hmm. So I have added all the references to you. You can just go read and see if it's clear, because if I can discuss, but it'll get a bit advanced and beyond the scope of your understanding. That's what I understood. Otherwise, we can again have a discussion in the next live session if you want to, a bit more on details. Okay.
Okay.
So just let me just tell you that, see, if you see it here in this particular figure, so here your stationary phase is polar, okay, and your sample you're passing through it, and your mobile phase is the non-polar, that is hexane. So you pass your samples, and if your samples are non-polar, it will take more time. Okay, okay. Let's discuss the next question. So once that particular MSc student, like with continuation to the last question, he has opted for like affinity chromatography, got the elute, and he has performed this gel based then he has performed the SDS gel, and this particular Prakhar is this okay, Prakhar wants to understand the possible reasons why the eluent profile looks like this. So just look in this gel picture, and this is the ladder, and these are the different lanes which has wash, which has its lysate, which has the illusion that this is the final one, the illusion, the ninth lane, if you see. Now if you look into this profile, what is the first thing that comes to your mind? Like if we are just focusing on the 63 kilodalton, this one, what is the first thing that comes to your mind? Anyone?
Then by looking at the ladder, we can predict the weight of the proteins in kilodaltons.
Yes, that's fine. So, okay, that's okay. That SDS page we are doing just to determine the molecular weight of the particular protein that has been eluted. But you see, there's not just one clear band. There are multiple bands in that particular range, right? So, any idea why has this happened, and why don't we? Ideally speaking, we should have a single band in every column if it is of the same protein. But why is this divided, or these multiple bands present along in the same line? Any idea? Okay, let's see what's in the chat box. Difference in band across reds, very good. Difference in bands. Now, why? You see that different bands, 1 2 1 2 here. Ideally, it should be like a single separate band like this, or maybe this one. Anyway, so out of these four options, the answer will be A and C. Okay. Uh, A is like the N-terminal histag might lead to production of truncated proteins. Now, truncated means short versions of proteins or the ones that has broken down. And C is bacterial protein has high histidine content. So these two options could be the reason. And why not multimeric proteins? Because that cannot be identified in this how multimeric proteins can be looked. For that, we need to conduct the native page, but this is, I guess, an SDS page. This is a non-native denaturing phase. And also, for the first two reasons, A and C. Now, this question comes with the query question that you said, protease inhibitors, right? Generally, when we are isolating proteins, in what we do is for isolating proteins, we treat the cells with lysis buffer so that the cells slice and the proteins come into the solution. And it's very necessary to add protease inhibitors to it so that your protein of interest or any XYZ protein is not actually degraded, because if it is degraded or denatured, it will like form multiple bands. So just to avoid the degradation of proteins, we have to use protease inhibitors. So is that clear? Somebody has that question, so that comes along with this question also. So we have to use protease inhibitors so that we don't have our product degraded. And here that is happening, something or the other has happened that has led to truncated proteins, and definitely the bacterial cell had high histidine content to it. Okay. Is it clear?
Yes.
So Prachi had a question that that about the proteins. Is Prachi there? Yeah, she's there. So she had a question about why we use protease inhibitors in it. So Prachi, is your question clear? Okay, great. So I just want to just make one thing clear. So if you have any doubts regarding any week sessions, like direct assignments, we cannot help you because that is something which you are assessed and like you need to submit that. But if you have any doubt on the contents or the specific topics, that please use the discussion forum that is in the main website of NPTEL, so that we are aware of the fact that okay, these topics are not clear to the students and we can just include that in our session. Like today we are this, because these questions had come up in the last forum we discussed. So kindly use it. And every Friday and Tuesdays we have two sessions to like a complete a 4-hour session, so we can definitely include that in the content. So just circulate this among others also. Okay. Okay, let's discuss the next question. Now, the next question says that if somebody wants to confirm each band, if it contains the desired protein of interest, like if we go back to this, he wants to confirm that all these bands have your protein of interest or not, what kind of technique should he be using so that to confirm? So just take a chance and like you can just give your answers in the chat box.
Then Western blotting.
Okay, and only Western blotting, or you think others also?
Mass spectrometry maybe.
Maybe. Why maybe and not confirmed? Okay, let's do something. Uh, Everybody, most of you are saying A. Okay, I mean, two of you are saying it's mass. So why isn't ELISA or microarray, any one of you, or Western blot? Somebody said Western blot, right? Instead of mass spectrometry. So I just want to know, why do you feel it's Western blot and not mass spectrometry? And the people who said mass spec, why don't you think that other three of them could be the result and just mass spec? Anyone? Okay, let's do something. Let's briefly discuss the different techniques that are mentioned here, and let's see that what could be the answer. Okay. Okay. The first option was mass spec. So mass spec is a broad process in which what is done is we take samples. It could be blood, tissue, even cells, animal cells, or bacterial cells. We digest it with trypsin, and then we subject it to something called the LCMS, which gives you peaks. So what happens is your digested proteins now have become peptides. Then we ionize these peptides, and each fragment gives you one peak. Now in the database, the idea of all peaks are already there. Like, okay, this protein has this number of peaks, or a particular protein has this number of peptides, or maybe if 10 peptides make one protein, 10 peptides are capable of making this protein. So all these kind of information is there in the database. What you need to do, you need to run your samples, you need to conduct a mass spec, and you need to see that your peaks match with the database or not. That is completely done by automated, it's done by the computer, and that will give you idea of all the proteins that are present within your sample. Okay, that is what a mass spec does. You find your peptides in the form of these peaks or like these peaks, and then you do a data analysis. That is a mass spec. So in proteome level, if you want to see what proteins are there, or not even in proteome level, only in a particular, maybe somebody is diseased and somebody is not diseased, you need to see what kind of proteins are present in the diseased person and what kind of proteins are present in the micros arrays. Okay, I'm just checking the chat box also in between, so I might just stop for a while. Okay, so for that you need the mass spec. Mass spec will give you a brilliant results. Now with Western blotting, what happens in Western blotting? Here also we are able to detect the unknown proteins or the unknown. What happens is you just load your protein on it, you run a gel like a normal SDS gel, that we polyacrylamide gel that we do, and then you transfer it on a membrane and you block it with a blocking solution or a buffer, that so that your protein of interest is not blocked, rest of the proteins are blocked. Then you add an antibody which has a detecting machinery added to it or a signal added to it, and that particular antibody is a complementary to your protein of interest that you had loaded in this gel. So it will definitely bind to it, and then you just wash and detect, like, because that has a signal attached to it, then you add that anything that will actually bloom out the signal and you will get the results. So that is what happens in case of Western blot. Now talking of microarrays, somebody wrote microarrays are used for gene expression studies. You're completely correct, but now protein microarrays are also there, where your protein of interest is already, or the total protein is already immobilized into a microtiter plate or in the form of a chip. Like you see, these are chip and it has proteins in it. Then you add antibodies which will bind to your protein of interest, which are conjugated with a detection signal, wash and dry, and you have it entire thing. That these are proteins. Dots are protein which has the proteins. These are antibodies that might possibly bind to your sample, and then the fluorescent signals. Mostly fluorescent signals are now used as detection system. And the fourth process is ELISA. ELISA, what we do? ELISA, generally we put an antigen on a microtiter plate, we add an antibody, we add substrate, and we also, there are many times, to just be double sure of it, we add double antibody to it, like a secondary antibody which is which binds to the primary antibody. So primary antibody is the antibody which first binds to it, and the secondary antibody is the one that will come second in line, and then we add the substrate, then you have a detection like system to it. So these are the 4 different techniques in very brief. So all these techniques seem same. If just after saying everything, if I was in your place, I would have thought that okay, these all sound same. But with the scope of this question, the answer is mass spec and Western blot, because these are the most sensitive and specific techniques out of all of these. And that is why for here, these 2 techniques are mostly used when we have to detect an unknown. And also one more thing, which is very important when it comes to detecting proteins or in proteomics, is high throughput. That at one particular, within a single reaction, how many proteins or how many samples can be analyzed together, or how many proteins you can detect together. For ELISA and these things, only single proteins can be analyzed. Like it's not like multivariate. So you need to identify many peptides or many proteins at the same time. So for that, Western blots and mass spec is very much important. So am I clear?
Yes, ma'am. Ma'am, there's a question in assignment 3 which is related to microarrays. Can you explain
Which
that
one?
question? Ma'am, should I type in the chat box?
Sure. Okay. What were the options?
And the options are he can extract total RNA from the samples and label and hybridize on the slide.
Hmm.
The other option was he can extract total DNA from the
Hmm.
samples and label and hybridize on the slide.
Hmm.
The other option was he can spot cDNA on array, extract total RNA from the samples, label and hybridize on the slide.
Hmm.
And the last option was he can spot either of DNA or RNA on the slide.
So what do you feel should be the answer?
I could not understand this because I don't know much about microarrays. So maybe
Okay.
you will
Okay.
explain that.
Okay. So I can just briefly explain something about the microarrays. That is, just give me one moment.
Ma'am, and the assignment's date was also passed, so you can answer because it was already submitted.
Okay. That I can do. That's what I was checking, the assignment dates. That's fine. I can
It
do.
was 15th of February.
Okay. I'll do it. No problem. Give me one. Just give me one moment, okay? Just one. So you got it correct or wrong? Just tell me this first.
My answer was not correct.
Okay, your answer was
I
not correct.
selected second option, and the answer was something else.
Okay. So you gave, like you told that you want to extract the DNA from the samples, label and hybridize it on the slide, right?
Yes, ma'am. I chose second option,
You
and the
chose
answer
DNA.
was
But I guess generally microarrays are done with cDNA.
okay.
That's my understanding, that RNAs are like microarrays on like for checking the gene expression are done on DNAs, because generally we extract the RNA from the samples, label and use a probe against it. So what is the purpose behind all these techniques is that your plate will have all kinds of genes in it. Now you need to find if your sample has those genes which are present here or not, right? So what we do is we take the sample maybe from blood, we extract the RNA, we convert it to cDNA, just as in expression vectors we were saying, and then we use it as a probe against the array of different genes that you have within the plate. That is what we do. So in case of that, you need to use a cDNA instead of a DNA. Is my point clear to all of you?
Yes, ma'am.
Okay. Okay. Oh, let's get back to where we were. Hmm. Hmm. Is my screen visible?
Yes, ma'am.
Okay. Okay. Prachi, is it clear to you? You chose first option. So I guess first option was something about RNA, just normal RNA or DNA. I don't remember the options. Okay. Let's get to the next question. Hmm. So, so again, after he had conducted the chromatography, he came to know that okay, his bands are near about 10 kilodaltons. That we were discussing that it corresponded to the different. The ladders are actually your references to which you need to see that okay, the if a band falls within this reference, okay, that means that my protein of interest is of this molecular weight. Okay, so he has done that and has found that his protein of interest falls within at least 10 kilodaltons approximately. So, which chromatography method now we should use to obtain the pure protein? Okay, we have 10 minutes left in this session, so you need to just leave and rejoin once again. We'll finish this question and then you guys can leave and rejoin. Okay. So, what kind of technique should he use if he has idea about the size of the protein? Let's see. Okay, D, rest. D.
So then
Now tell me why.
if he wants to choose on the basis of size, that I think it should be size exclusion chromatography.
So, what does a size exclusion chromatography do? Like, what is its principle?
Mm, it will separate proteins on the basis of their different sizes.
Okay. So, suppose this particular protein is 10 kilodaltons and you had one more protein which was 11 kilodaltons. So, will that be eluted along with this or separately?
It
Suppose
will
you have
depend on
three different proteins. One which is 10 kilodalton, it is your histidine tag, that protein. One protein you have of 8 kilodaltons and another protein you have 12 kilodaltons. So, what will be the elution pattern? Which one will come first?
11 kilodaltons.
Good. Who said that?
Nidhi.
Okay, Nidhi. You're correct. So the concept behind size exclusion is that if you know the size, you can. It's again, if you know the tag, you can use affinity towards it. If you know the size, you can use a porous bead according to that size. So yes, it's size exclusion. So we're done with 5 questions. Is there any other question from the assignment or any place that you feel like you are stuck on and you need some help regarding the clarity?
Ma'am, the 10th question of assignment, but it's not based on chromatography, it's based on something else.
That sum, a protein, the plasmid and DNA concentration sum.
Yes, we have to find the total yield of RN in microgram. 10th question.
Okay. I'll discuss today and you'll get idea of what it is. So I'll just discuss it. Maybe we'll not solve it together today, but we have a same question and you'll get complete hints to it. Just solve it after the session and post it in the discussion forum. Were you able to solve it or not? If not, then again on Monday we'll discuss that. Maybe Anubhav will discuss that. Okay. We have one more question like that today. Anything else? No, I guess. Okay, let's go to the next question. Yeah, so it was size exclusion chromatography. Correct. Okay, now, so are you aware of Sanger sequencing, all of you? Do you know what is sequencing? Firstly, what sequencing is? What is the purpose of sequencing? And why we need Sanger sequencing? Like, what Sanger sequencing, why we do Sanger sequencing? Does anyone have any clue about it? Okay, let's do. So sequencing is basically you have a DNA sequence. You got a DNA sequence, and now you want to know, you got a DNA actually. You purified a DNA from your sample and you want to know what is the sequence of it. Suppose you identify a new bacteria or maybe a new virus particle. And now you want to know what is the genetic makeup of it. For that, what do you need to do? You need to first isolate its gene. You need to then see that what is it made up of, what is exact sequence of it for your downstream experimental purpose. So in that Sanger sequencing is one of the sequencing techniques which utilizes the concept of chain termination sequencing. Now, what happens in sequencing is that suppose you have this is your template strand, this is your main strand, or maybe you can see this picture, this is a template strand. You actually synthesize one more strand complementary to it and you keep synthesizing it. In that you keep on like providing the different nucleotides to it and it will be synthesized. Definitely a small primer and a polymerase is given in that so that it keeps on synthesizing, and then at the end you will get a complementary strand to it, and in that way you know, okay, the sequence was this one. But this cannot be done infinitely, like you cannot keep on synthesizing. So what method is used in Sanger sequencing? In place of DNTPs, like normal DNTPs, that is the nucleotides, the you use a di-deoxynucleotide. In generally, what we use the deoxynucleotide, which has a OH group in the third position. You see, this is your base, and you have a OH group in the third position. But a di-deoxynucleotide has a H group in the third position. Now, why is this third position very important? Can somebody say? In case of DNA or in RNA, why is the 3' OH very important? What is the role of this 3' OH? Anyone?
It forms the phosphodiester bond between.
Exactly. So, to elongate a particular strand, what happens? 5' to 3' direction, right? So, the phosphate group of the previous base will actually form a phosphodiester bond with this one. I mean, this particular previous base, I mean this base to join with the next base forms a phosphodiester bond, and this bond is between the 5' OH and this one. 5' phosphate group and the 3' OH. That is how the structure of the DNA is formed. So now, if for some reason we replace this OH group with a H group, the synthesis stops. And so does the strand become only that, like it cannot be elongated anymore and it stalls at that portion. So now this concept is used in Sanger sequencing. What happens is, suppose this is your main sequence. Okay. You provide it with normal mixture of DNTPs as well as with DDNTPs, which are fluorescently labeled. Now, if something is fluorescently labeled and you excite it at a particular wavelength, it emits at another wavelength and that gives you a light or a signal. That is detected by a detection system. So now, if a DDNTP is labeled with fluorescence and is supplied to this in some portion, suppose like you see GATG. So something against T is A. If this A comes and binds here, after that nothing will be synthesized because that particular A bound to T, that is the end point of it, and that will give you a signal. And again, maybe when again a new cycle starts, it stops at G. That is, it will stop at C. It will give you another. So what is generally done is 4 different tubes are used. Each tube will have the template strand, all the DNTPs, and one fluorescently labeled, maybe a G, a T, a C, or A. Like this, you will have 4 different aliquots which will have D, T, G, C. And as a result, what will happen? That you will have different fragments which are ending at the same thing. So like that, when all the fragments will accumulate, you will find a chromatogram like this, like GAT, like this. So these are the intensity, like maybe you had more T's of the same size, a little bigger, and you had less T's, less strands ending with T. So this is how the chromatogram will look, and you will know that okay, this was my result, or this is my sequence. Is it clear how a Sanger sequence is done, or you need more clarity?
Yeah, ma'am.
Clear, okay. So that is how you do.
Mm.
So if in this question, yes, anyone was saying something? Oh, they are added at the same time. Everything is added. The entire reaction mix is added, and now it is completely automated. I have actually provided a link in here. It's from Sigma. You can just go and see how it is exactly done. So everything is automated now. You just need to put your reaction mixture and it will detect. So what happens is, like, these are all you see. Now you have these fragments, large fragments, which have completely synthesized and ended at the G. So these small fragments, all these were fluorescently labeled. Now there's a laser beam. It will detect the fluorescently labeled T's. Normal T's it will not detect. Only the labels which are only the tags which are labeled with fluorescent T's will detect it. So strands which end with normal T and strands which ends with DDNTP, which has T, are not the same in terms of fluorescence signal. That is how they are different. So what will be the answer in this? Which he needs to confirm that the sequence of the recombinant plasmid he prepared. He used Sanger sequencing of DNA molecules. Which of the following is correct for the sequencing method? Okay. So what will be the answer? Somebody, any one of you?
Ma'am, see, because as you explained just that it lacks DDNTPs lacks three prime hydroxyl groups. So because of its absence, there will be termination of DNA strand synthesis, and hence the fluorescence will emerge.
Correct. Absolutely correct. Okay. So, okay. So is the concept clear for all of you?
Yes, ma'am.
Or the ones who just learned it need once, well, like it would be great if you want me to explain it once again. Okay, clear. Let's go to the next question. Okay, I guess Nidhi, no, she was asking me about this question regarding the sum.
Ma'am, I was asking Priyanka.
Priyanka was asking. Okay, Priyanka. So now what is the concept behind this sum? Okay, let's do. Okay. So the thing is that let me first describe this question, and then you can just post it your question in the chat box, and I'll just go through it and I'll help you in solving it. Okay. So
Okay.
so he isolated a recombinant plasmid for sequencing and diluted the sample. Hmm. And he diluted the sample. Hmm. He diluted the sample by factor of 1:40. Then the spectrophotometer reading was at 260 and 280 nanometers. That was respectively 0.42 and 0.21 respectively. So to calculate the plasmid concentration in the dilute and concentrated plasmid samples, how do we do it? So was the new question like this or a bit different? Priyanka, I guess. It asks for
Quite
RNA yield.
similar.
Okay, quite similar.
It asks for RNA yield
Okay.
in micrograms.
So let's just break down the entire question. First thing is a dilution factor. So are you Aware of what is a dilution factor and how it is done? Any one of you? Do you know how we dilute a sample and how this what is the concept behind a dilution factor? Okay, so I'll just begin. Dilution factors are basically that when you have a sample and you want to analyze, you cannot, generally what happens is you have the crude sample or the isolated and purified sample. You cannot utilize the entire sample for one particular experiment. You need to store it. So generally we dilute it, like maybe half diluted, one third diluted, one fourth diluted, like this we dilute. And by what we mean that if it's half dilution, one part is sample and one part is the diluent or the solution in which you are diluting. So your dilution factor stands as 2. So if it's a fourfold dilution, it's a fourfold dilution. It's like 1 part sample and 3 part diluent. Then the total volume stands up to be 4. It's actually how we calculate if the final volume of your solution by the initial amount of sample taken. So here is it like if it was half fold dilution or two-fold dilution, it would be 1 part, the final volume is like 2. So you had 1 part of your sample and you added 1 part of the dilution diluent, that makes up total 1 plus 1 2, that is a final volume. So it is 2 is the final dilution or the fold dilution. So generally what we do after we get the results, we multiply it because you started your experiment with a diluted sample, so you need to multiply the dilution factor with it to get the final concentration. So similar thing is asked here. So it was diluted by 1 by 40, where 40 is the final volume. Okay. So your UV visible spectrophotometer. So in order to detect a plasmid, protein or DNA, one method is a spectrophotometer. Why? Because at different wavelengths, you get a peak of it. So it is seen that in case of DNA, we generally use 260 and 280 nanometers for like DNA and for protein. So can anyone say, but for DNA, why is it 260 and for protein, why is 280 nanometers? Like why this specific wavelengths are not something else? Can anyone, does anyone have an idea why this wavelengths are there? Anyone?
So the absorption may differ for DNA, RNA, protein and all.
Exactly. So we need a absorption which is maximum for DNA at a particular wavelength. Then we can say, okay, DNA absorbs at this wavelength. So for DNA, it's 260 and for protein, it's 280. So that is what he did. He did for both, for protein. And now I'll come to the point why he has done it for protein at the end of the question. Like why protein? He just wanted DNA. So he isolated protein like DNA, plasmid DNA, and then he could have just performed it at 260 and done his experiment. But no, he has done it with protein also. Now we need to calculate the plasmid concentration both in the diluted sample and also in the concentrated plasmid sample. For this, you need to have an idea that what does the absorbance correspond to. Like if your absorbance is 1 at 260 nanometer, then it corresponds to 50 microgram per ml of DNA. This is a standard. The same standard comes for RNA and for protein. So this is the main thing that we use. Like if 1 OD corresponds to 50, then how much was your OD here? 0.42. Then your 0.42 OD corresponds to 21 microgram per ml. Now let me get through the calculations. 180, 50 microgram per ml. So nanogram per microliter or 1 nanogram per microliter is also 1 microgram per ml. So here, because everything it's actually the same. If you see, like 1 nanogram per microliter is 1 microgram per ml. So you just multiply 50 with it and you have this result. Okay. Now you need to find the concentration. For that concentration, you have to multiply with the dilution factor. What was your dilution factor? This is your concentrated, like
40.
1 by 40, exactly. So you multiply it with 1 by 40, you have 0.52 microgram per ml. Okay. Correct? So that will be your option
D.
D. Now, why did I include this one? Initially, when I was solving it, I found this is also the same nanogram per ml. Why is it different? here comes in the catch that nanogram per microliter, and here is nanogram per ml. You need to be very careful about these units. Okay. Now, is it clear for all, or should I repeat how to solve this question?
Ma'am, it's clear to me.
It's clear to you and to your friends?
Clear, ma'am.
Clear, right?
Yes, ma'am, clear.
So the same thing will come for DNA, RNA, and protein. Like what does one OD at 280 unit or 260 unit, or for RNA, how much that does quantify as RNA? So Priyanka, you just go search in Google that what does one OD of unit correspond to that, and I guess we'll be able to solve it in that way. Have you tried it in that way, or like you haven't?
Ma'am, I tried. I will try it again after you've explained me. Now I will try it again.
Okay, for Supriya, I'll explain it once. I'll just try one thing. I'll just try to share a new screen where I can just solve it for her. Just give me 1 second, all of you. So is my screen visible? Like I'll be just solving the sum here. Is it visible?
Yes, ma'am.
Yes, ma'am.
Okay, the second screen, right? So
Yes,
what
ma'am.
was the absorbance? The absorbance at A260 nanometer was 0.4142, right? I'm just writing it in a second screen. So are you guys able to see it?
Yes, ma'am. Yes, ma'am.
Okay. Okay. So the question was at this, and the dilution factor used here was 1:40. Correct?
Yes, ma'am.
Okay. Hmm. So the question is, we need to find the concentration of the plasmid in general, and also the concentration of the diluted plasmid solution. So what is the convention that we spoke of? That in case of DNA, 1 OD at 260 nanometer corresponds to 50 microgram per ml. Okay, so now I have 0.42 OD that is given in the question. Just multiply 0.42 into 50. That will give me how much? 21 microgram.
21.
Yes, per ml. Now this is my concentrated one. Now I had diluted it by 140, 1 by 40, not 140, by 40. So I just need to multiply this into 1 by 40. This is basically the division of it. So it will give you 50 microgram per ml. Be very careful of the units. Now, one more thing that is mentioned in this one, like the table, that is, we also check the purity. That is why the two things are given here, the two readings. So DNA gives a major peak at 260 nanometers. Protein gives a major peak at 280 nanometers.
280 nanometers.
So if we just, you will always find these experiments have the OD at these two values. So if you just divide it and your value is 1.8, it means in terms of purity, it has DNA. Okay. If your value is 2 and greater than equals to 2, that means you have RNA in it. And if you have lesser than 0.1 or very specific lesser than 1.8 or specifically like around 0.6, then you have a protein contamination rate or protein rate. So many a times after isolating DNA, what we do is we try to see that they have contamination or they don't have contamination. Okay, that is how we select it, just to check the purity. So generally, I have done this by my hand. Like we have checked after isolating plasmid, and like this was my third year practical. Actually, we had this practical molecular biology practical in my graduation course. So there we were given like we have to isolate plasmid and then run gels and then check that whatever sample we have isolated, it is correct or not, like it was pure or not. So that is how we do it. Okay. So is this clear?
Yes, ma'am.
Okay. So let's go to the next question. So are my slides visible once again?
Yes, ma'am.
Okay. So it's D. Okay. We are almost at the end of the session, like questions. And so this is again a kind of conceptual question where we need to think that what is happening here. And so a researcher group has decided to use a different technique in order to achieve an intact insulin as a dietary supplement. So dietary supplement, keep that in mind. So dietary supplement means that it could be just supplemented through diet. Okay. So what did it do? By diet means that, okay, let me complete the question first. They succeeded in obtaining the desired protein in a goat's milk. Which of the following is true for the above mentioned condition? So, suppose somebody takes insulin. So, what are the different forms of insulin intake that is now present? It's prescribed. One is like direct injection that they do, and there are tablets of insulin, and there are tablets of injectable tablets are there, right? And it's painful. It's not very easy for the people, diabetic people, to like keep on in injecting insulin every day. Even kids also have that. So it's very difficult. So scientists are trying to find that if in any way we can supply insulin through diet. Like suppose we isolate insulin from goat milk and we feed that milk to a diabetic patient. Will that insulin be, can that process be utilized? Like if so, suppose that process can is a good successful process. So what was actually done to get insulin out of goat's milk? So is the question clear? You can just have a guess that what could be the answer here. A.
Ma'am, the goat is a transgenic and a gene of insulin is added into the genome of the goat because there was the desired protein was present in the goat's milk. So it is possible only if we make the goat transgenic and introduce the gene of interest.
Okay,
Only then
so
should be able to produce the desired protein.
good. So you mean that you create a goat that has whenever milk is secreted, or the cells that are secreting milk, that will have insulin gene within it, and the product milk will actually have insulin within it, right? That is how the goat was designed, to produce insulin in its milk. True? That's what you
Yes,
mean.
ma'am.
And rest of you? A. Okay. Good. Yes, A is
A.
the answer. Yeah, A is the answer. Correct answer is A. You just design a goat that is capable of producing insulin in its milk. So for that, you need to insert your gene of interest insulin within the goat, and that will be a transgenic. Now, you have to specifically insert this gene so that it expresses in the mammary glands, not throughout the body. Obviously, these are very like specific insertions that take place. If we insert it in such a way that all the cells will have, but it will be of no use. You cannot extract that because milk is a secretory product. We have to aim that, okay, we are going to insert insulin in a particular gene so that ultimately it's expressed in these kind of cells which are present in the mammary glands, and milk is produced in that way. Is it clear? That is how actually transgenic animals are now being used. They are trying to different drugs and through dietary supplements. Like this, there is this different plants like we have, like There are different plants. I don't remember example right now, but there are plants which acts as dietary supplements to other like drugs or important nutrients which are normally not like provided or normally is not acquired by human or has a deficiency. So on consuming those plants, those particular drugs or supplements can be like reached, or it can be like subjected to the patient, and he or she can have that particular supplement by consuming those plants or fruits in that way. So is it clear that how a transgenic animal is produced?
Yes, ma'am.
Okay. Okay, the final question. Many in the discussion forum, we found many questions related to RNAs and RNA isolation. So, oh, I already showed things. Okay, now just tell me that, just do a match the following and then tell me that what could be the thing, like what will be the possible explanation along with the answer. I have any one of you tried answering this? Okay, let's go by one by
Ma'am,
one. Like,
uh,
oh yeah, sure,
ma'am,
sure.
can we have option C as answer?
Yes, and why?
Ma'am, because DNA is used to degrade the genomic DNA contamination. So for
Correct.
C would be the match. And for RNA ZAP, it is used to prevent RNA degrading when we try to isolate the RNA.
Good, very good.
So that's why I've selected the option C.
Okay. And EDT and trisol we'll discuss. Good, very good. So RNA zap. This is RNA zap. So RNase is an enzyme which degrades RNA. So we need to inhibit the activity of any RNase or protein present within our sample. So RNase is a contaminant. Actually, it contaminates, and there are RNAs present everywhere. Even in the palm of your hands, you will have it. So if your RNA that you're trying to isolate gets degraded due to these RNases, then how will you isolate? So for that, you need to prevent this RNase from degrading your sample of your RNA. So for that, this RNA ZAP is used. So which prevents degrading the RNA. That is, it actually prevents RNase activity, which will actually prevent the RNA from degrading. So in a nutshell, it prevents the RNA degradation. Okay. B was EDTA. EDTA, we all know that is a chelating agent, which is hugely used. Chelating agent. Now it chelates the magnesium and calcium ions. We know, and DNA polymerase. DNA polymerase uses these two ions, magnesium and calcium, for its activity. Now, if we are able to inhibit the DNA polymerase activity with the use of EDT, so automatically my DNA will be degraded. The Now, while isolating RNA and DNA from a sample, like specifically isolating RNA, many a times DNA also comes within it. So we need to like remove that so that any kind of DNA contamination is not in the sample, and ultimately we have a pure sample with only RNA. So EDTA is used to degrade the genomic DNA. Just one second. B5 prevents harm, and also it prevents RNA hydrolysis. So this is the role of EDTA. Now EDTA is both important in terms of DNA also, in RNA also, but here it's specifically if we try to map. So this is one role of EDTA. The second role of EDTA is it prevents RNA hydrolysis. So again, we have to, our aim is to protect the RNA. So how to protect, so it by any chance it shouldn't be hydrolyzed. So all the hydrolyzing enzymes, the way we mentioned that it chelates the important ions that are used by these hydrolysis enzymes. So that is how EDTA is helped. See, DNAs, I've already discussed that how DNAs is, if we DNAs degrades the genomic DNA contamination, it directly like degrades the DNA. DNA is it's very simple thing that DNAs will degrade the DNA. Anything that terms with A's, RNAs, DNA A's, like ASE,
Ma'am,
it degrades. Yes.
ma'am, I have a doubt. Like, EDTA, don't we use that in DNA isolation?
Yes, we do. Why do we use it? What I mentioned, it is a chelator. It chelates the ions, right? So while DNA isolation, we need to break the cell walls so that the and the cell walls have these ions, so it chelates the ion. So what happens is it removes the ions and the cell walls break and the DNA comes out of it. So that is also one of the roles of EDTA as a chelator. Is my
But
point
ma'am,
clear?
you here said like it will inhibit the DNA polymerase. Like,
Yeah.
can you explain that again? I didn't understand.
So the thing is that what happens is we want we don't want any DNA, right? The polymerase. So how is DNA formed? DNA polymerase is formed. Like what is the role of DNA polymerase? To synthesize DNA. So we need to stop this process. And this DNA polymerase requires magnesium and calcium ions in it. If you see the enzymatic action, calcium and magnesium ions or different ions are required for functioning of the polymerase. And if those ions are not present, then polymerase cannot function. Now, if we use EDTA to it, these will chelate these ions that remove that ion and form a complex. As a result, polymerase will not form, and what will happen? DNA will be degraded. Like not degraded, but it will not synthesize only in that point. So that is how EDTA will function in this way also. Clear? Is it clear? How, what is its role in DNA polymerase, EDTA? Clear? Now, third is DNAs, as we described. DNAs is, it degrades the genomic DNA. Yes, ma'am. Okay. And the fourth one is the trizol. Trizol is actually used for lysing the cells and bringing out its content. So what we do is we add lysol directly to the cells and we centrifuge it. So it forms like a, it forms two phases, and in which the DNA and the all the cells are burst out and the DNA and RNA comes into the solution. So that is the role of trizol here. So are the roles clear?
Yes, ma'am.
Yes, ma'am.
So with that, we come to the end of the question answers, and I would request you all to like put one or two questions on yourself so that we can like discuss that. If you have any doubts or like any particular topic, if you need, we can just briefly describe or discuss what you understand on. Anyone? Is that concept clear that what is the role of EDTA and DNAs and DNA polymerase? Okay, good. I'll answer that. Like how EDTA actually helps in D DNA and RNA extraction, what is its role? Like, just remember one thing that in case of whenever we are isolating RNA or DNA, we need these two things intact. Any kind of A's, like be DNAs or RNAs, can actually degrade your sample DNA or RNA. So we have to prevent these enzymes, and these enzymes only active with using some metal ions. And if these metal ions are removed, that is by EDTA, then they cannot function and your samples remain intact. So that is how we are keeping it intact. You're using EDTA. Okay. Now let me come to this question. Experiment where cDNA synthesis was performed, incubated overnight with isopropanol. Okay, okay, okay. So DNA extraction is mostly done using aqueous phase isolation technique, that is known as with isopropanol. So what happens is uh, it divides the solution into phases, and when you're using this alcohol isopropanol, so you can isolate it directly from that. That is why we keep it on. Now the incubation time differs, like you keep it overnight on isopropanol and then you extract the DNA the next day when you come. So these solutions actually isolate your DNA, and when you add isopropanol to it, your DNA gets separated from rest of the solution, and from that phase you can collect the DNA. And again, when we are talking about using isopropanol or like DNA, we have to keep in mind the charge. Like DNA is negatively charged, so you have to like negatively charged. So in order to help it precipitate, you need to neutralize it so that it precipitates with the solution. So when we are using isopropanol, my understanding is isopropanol is DNA doesn't dissolve well in isopropanol. So it easily it will not dissolve and it will precipitate. And so that at low temperatures it will precipitate, and the next day you can just take it from the below portion of it. That is how it's used. Prachi, is it slightly clear to you? So okay, if we don't have any other questions, anyone? So just for your like how I understood these concept is you can just go to Thermo Fisher or Sigma, these different sites who actually make these solutions and they provide commercially. You can go to the site and their technical handbooks, and you can see the role of these things, like why isopropanol, while RNAs. Anyway, in papers you will get it, or in books also. But they give very good illustrations and they provide that why these are used. So you can go there and just see that what is the main concept behind using particular thing in a extraction protocol. Okay, so with that we come to the end of this session, and I guess I can end the session if you have no other questions. We still have 7 or 8 minutes left for the session to get over. So if any doubts or any questions, I'm ready to answer them. Anything? So we'll see you next Friday. And again, we'll be discussing week 4 questions. If anything, that any topic or any concept you want, please feel free to discuss or just post it in the discussion forum and also in the Telegram group if you feel like. And I hope that all of you have joined the Telegram group. If not, then NPTEL has sent an official mail to all of you regarding the group joining details and all. So feel free to interact with us. And I hope that the session was enriching field. Okay, thank you.
Yes, ma'am.
Thank
Thank you, ma'am.
Thank you so
Thank
much, ma'am.